Good day, everyone. Today, I will be discussing the process of DNA replication, the method by which cells make an exact copy of their DNA before cell division. What is DNA? DNA is a molecule that carries our genetic information. So every time a cell divides, it must pass on a complete copy of its DNA to a new daughter cell. So this is made possible through a biochemical process called DNA replication. So the key idea for this is behind the replication is base pairing. So the rule that adenine pairs with thymine and cytosine pairs with guanine. So that is the one idea behind the replication. So next is a step-by-step process of DNA replication. So first step is the unwinding by DNA helicase. So the enzyme DNA helicase attaches to the DNA molecules and begins to unwind the double helix. So as it moves along the strand, so it breaks the hydrogen bond between the base pair, like mura siyang mag-unzipping the zipper. So it forms Y, like they create a Y-shaped structure known as the replication fork. So where the DNA continues to separate. So this step is like unzipping the zipper, like it is a shape. It is make a shape. It is a Y shape that is called replication fork. So second is the base pairing with free nucleotide. Once the strands are separated, the exposed base attract free nucleotide found within the cell. So cytosine pairs with guanine and adenine pairs with thymine. So each nucleotide attaches one at a time. So once a nucleotide pairs with a template base, another enzyme called DNA polymerase. So DNA polymerase is to check if pairing is correct. So if correct, the DNA polymerase forms a new phosphoester bond. So it is connecting the nucleotide to the growing DNA strand. Third is the formation of leading and lagging strands. So because the two strands of DNA run in opposite direction, one from 5 to 3 and other from 3 to 5. So DNA polymerase can only work continuous on one side. So the strand that grows continuously as to 5 to 3 direction is called the leading strand. So the other strand, which grows slowly in short segment, is the lagging strand. If maguna siya sa 5 to 3, it is called leading strand. But if 3 to 5, lagging strand. So these short segments are known as Okazaki fragments. Okazaki fragments. So gaps between these fragments are eventually joined by an enzyme called DNA ligase. So sealing the strand into a continuous new DNA molecule. So next is a DNA replication at multiple sites. So DNA replication does not start at just one location. Instead, it begins multiple regions of replication along the DNA molecule. Next is the result. The end result of DNA replication is the formation of two identical daughter DNA molecules. So each daughter molecule contains one original strand and one newly synthesized strand. These two DNA copies ensure that when the cell divides, each new cell receives the complete genetic information it if needs. is the DNA and chromosome. So after DNA replicated, it coils tightly around special protein called histone. So this forms structure known as chromosomes. So each chromosome is combination of DNA and proteins, so helping organize and stabilizing the long DNA molecules. So the last one is the conclusion. So in summary, DNA replication is fully coordinated with these five. First is the unwinding the helix of DNA helicase. Second is the base pairing using free nucleotide. And the third one is the assembly and proofreading by DNA polymerase. The fourth one is joining of fragments by DNA ligase. And the last one is the multiple replication origin for efficiency. So this ensures that every new cell receive a perfect DNA. So maintaining of continuity. It is maintaining continuity of our life. So that will be all. Thank you for listening.